Dear students, today we are going to study pathogenesis by bacteria and the defense mechanism which is offered by our body in response to the bacterial infection. First of all, in pathogenesis, we'll take up host-parasite relationship. These are certain terminologies and aspects in bacterial pathogenesis which we need to know. Let us take up benign parasites. These are those parasites which could not able to develop disease in the body because of the strong resistance which is offered by the body renders them a bit ineffective, and they are unable to cause disease in the host. Therefore, they are forced to create a relationship which is benign, and they do not cause any symptoms. Only subclinical or latent, the bacteria remains latent and the symptoms are subclinical. But however, the parasite drains nutrition indefinitely as long as it remains in the body. Inept parasites are those parasites which overpowers our proposed defense mechanism and we develop disease. Such parasites which develops disease are called as inept parasites. There are some parasites which are opportunist. They are part of normal microflora of our body, but whenever they encounter certain conditions, they tend to develop disease in our body. Diplococcus pneumoniae is such an example, which inhabits the pharyngeal region of our throats, but whenever we encounter very cold conditions, we develop the pneumonia. Now let us come to pathogenesis. Antigens. What are antigens? Antigens are actually the chemical substances, or they may be a capsule of the bacteria, or capsids of the virus, or cell wall of the bacteria, or the flagella of the bacteria. Aggressins are extracellular enzymes having the ability to break cells and connective tissues. Now there are certain toxins which are released by the pathogens. Some of them are exotoxic toxins and some of them they are endotoxins. Exotoxins are normally produced by gram-positive bacteria, whereas endotoxins are produced by gram-negative bacteria in general. The nature of exotoxins is normally proteins, whereas that of endotoxins it is lipopolysaccharide. Even the small dose of exotoxins is lethal to us, whereas endotoxins generally we require a large amount of large dose to be lethal. The exotoxins, they are heat labile, that means they are easily inactivated by the rising of temperature, whereas endotoxins, they have a high tolerance capacity to heat, and therefore they are very heat stable. Exotoxins, they are sensitive to temperature, alcohol and formaldehyde, whereas endotoxins, they are sensitive to alcohol and dilute acids. Upon denaturation, the exotoxins are non-toxic, and toxoid can be prepared with the immunogenic properties from the toxoids. Then neutralization of with the antitoxin is not possible with the endotoxins. Now let us take up some examples of bacteria which secretes exotoxins. Bacillus anthracis, Diplococcus pneumoniae. Then we have Clostridium tetani, Clostridium perfringens, Clostridium tuberculei, Clostridium botulinum are some of the bacteria which secretes the exotoxins, whereas endotoxins are produced by Bordetella pertussis, Shigella, Salmonella typhi, Vibrio cholera, Neisseria gonorrhoeae, and Neisseria meningitidis. Now, after infection, the host body necessarily offers some resistance through activation of its defense mechanism. We have two kinds of defense mechanism, non-specific and specific. Now let us study what is non-specific kind of defense mechanism exists in our body. It involves immediate destruction of pathogen on the surface layer itself. It involves the mechanical defense like sweat, mucus glands, the pH change, peristalsis, the lacrimal gland which exists in our eyes, offers the mechanical defense. Next one is cellular defense. The phagocytic cell such as neutrophils and monocytes, they and microglial cells of nervous system, they just eat up the pathogens. Some more examples are Metchnikoff cells and Kupffer cells. Kupffer cells, they exist in liver, as you know, and these eat up the bacteria which comes through the blood supply in liver and thus cleanses the blood. Now we have lysozyme, as you know, is present in saliva, in tears. We have opsonins, which enhances the phagocytosis of the pathogens, and antibodies which develop due to infection. The antibodies along with pathogen, they get phagocytized, and this process is called as opsonization, and such a phenomena is triggered by opsonins present in the body. So first was mechanical defense, second was cellular defense, and in cellular defense we had seen that many types of cells which eats up or phagocytize the pathogens are present in our body. Opsonization is eating up of the pathogen along with the antibodies. Then we have physiological defense, in which we have agglutinins which immobilize the antigens by causing clumping. We have precipitins which converts the soluble antigen into insoluble ones, therefore inactivating it. We have antitoxins, combines with toxin to produce toxoid, and which neutralize its toxicity. We have lysins for lysis of the pathogens, and as I've told you right now, opsonins which enhances the phagocytosis. Fourth one is inflammatory defense. The pathogen releases leukocidin and hemolysins which destroyed RBC and WBCs. Now the host responds by converting soluble plasminogen into insoluble plasmin which dilates the blood vessel. That means this plasmin is a vasodilator. This renders the inflow of WBCs and the neutrophils, a kind of WBC, phagocytize the irritants or the pathogens. Then vasodilation is also caused by histamine and prostaglandins. This vasodilation renders or attracts more neutrophils to the site of infection along with monocytes, and the procedure they follow for reaching at the site of infection from the blood vessel is diapedesis. Fever is another mechanism of defense which is offered by host. The phagocytes due to presence of toxins releases interleukin 1, which is an endogenous pyrogen, and when these go to hypothalamus, which controls the temperature. The hypothalamus is induced to produce prostaglandins. These prostaglandins, they raises our body temperature, and this raising of body temperature discourages the presence of microbes in our body. Then we have antimicrobial substance in our body like serum protein complements, which are called as properdins, and we are also having interferons as antiviral components. So this was the non-specific defense offered by our body. And in the non-specific defense, let me revise. Firstly, we had studied mechanical defense, which is caused by sweat, offered by sweat, the mucus, the pH, high acidic environment which is present in our stomach discourages the bacteria and other pathogens which goes inside our body through food. Now thereafter we have cellular defense offered by many macrophages which eats up the pathogens. Then we have certain physiological defense mechanisms offered by our body. And then inflammatory response by histamine, which and other compounds which causes vasodilation renders enables our neutrophils and monocytes to just seep out into the site of infection. Another mechanism was which we had seen was fever, and the rising of fever, our body temperature discourages the survival of pathogen in our body. Then we have certain antimicrobial substances. Now let us come to specific defense. That means defense which is specifically meant for specific kind of antigen in our body. Specific counteracting of foreign organisms from production of antibodies and sensitized lymphocytes is called as specific defense. As you know, we have lymphocytes in our body. We have two kinds of lymphocytes in our body. One is B lymphocytes, another is T lymphocyte. T lymphocytes and B, there only one difference is there, that the B lymphocytes, they get matured in the bone marrow itself, whereas the T lymphocyte, they get matured in the thymus gland, which is present in our thoracic region. B lymphocytes are mainly involved in production of antibodies, and we have many kinds of immunity which is produced by production of antibodies against a certain and specific kind of antigen which our body gets encountered. Naturally acquired immunity is that immunity which develops when a person is exposed to antigens in daily life. That means before birth we don't have naturally acquired immunity. Another kind of immunity is naturally acquired passive immunity. This is developed by transfer of antibodies from mother to infants. Then we have artificially acquired immunity. How do we acquire artificial immunity? It is by vaccination. What is vaccine? It is generally the introduction of attenuated or weakened antigens into the individual, and in response to those weakened antigens we develop certain antibodies, and whenever we encounter the actual pathogen, we could able to offer a great resistance because of already developed antibodies. Now, artificially acquired passive immunity. How does this immunity develops? It is by injection of immune serum in susceptible individuals, like person bitten by snake is injected with antibodies from horse that is immunized to snake bite. Right now we are doing the same thing in COVID infection, that the person who is already infected donates his plasma which contains the antibody against the coronavirus, and if we inject the antibodies which were already developed in the infected person to the person who is suffering, therefore we can buttress his immune system against the fight of COVID. Now we have another system which is called as humoral immune system. Students, we are going to take up humor what is there in the humoral immune system? The humoral immunity is conferred through stem cells of bone marrow called as B cells. As I've told you in the beginning of this lecture, that humoral immune system is the antibodies, they are present in the humor or the fluidic part of our body. That's why this is called as humoral immune system. In this, the bone marrow cells produces specific antibodies in response to immunogenic organic molecules which are called as antigens, having specific regions as antigenic determinants. These antigenic determinants, they are identified by the antibodies or the B cells and produce antibodies against these antigens which are normally present on the surface of the pathogen. What are haptens? You will come across this term also. Haptens are low molecular weight antigens which functions unless attached, which don't do, let me change the slide. Which functions unless attached to a carrier molecules like serum protein. That means they are low molecular weight proteins. They do not function as antigens unless they are attached to a carrier molecule. If an antibody is formed against hapten, it will react with antigen even in absence of carrier molecule. For example, penicillin acts as hapten, which is not an antigen itself. Even then, some people develop allergy against it. Many people, they die of penicillin allergy. It is because of this. Antibodies belong to a class of proteins called as immunoglobulins. These antibodies, which are triggered or produced by the humoral immune system, they are of class of IgG or called as immunoglobulins. Now we have cell-mediated immune system. This involves specialized lymphocytes known as T cells which do not secrete antibodies but contain certain antigen receptors and attached to their surface. This immunity is more effective against bacteria and viruses because the cell-mediated immune system or the T cell, they actually are effective against intracellular parasites which are normally not secreting in the blood, but the cell which is infected has to be killed, and this is done by T cells or the cell mediated immune system by the help of major histocompatibility complex. So students, this is all in this lecture. In the next lecture we'll proceed further and we'll study epidemiology. Thank you very much.